Alrighty, so welcome to this lecture. We're going to go over some of the basic concepts about DNA structure and function, as well as RNA structure and function. Again, this is just a very general overview without going too far in the nitty-gritties of the genetics of the two topics. So we'll give this a go, starting with DNA. Now, DNA is a hereditary molecule, as we call it. It's the genetic material that organisms inherit from their parents. Each chromosome contains one long DNA molecule consisting of several hundred to thousands of genes. And in order for it to be hereditary, there's two characteristics that are necessary. First is stability of life, right? So you inherit genetic information or DNA from mom, and you also inherit it from dad, who both in return receive their genetic information from their own parents. Now, because it's inherited, it can be passed on through generations and thus be preserved or maintained. So you have the creation of species genetically. The second component or characteristic is mutagenic properties. It's important that there is some amount of variation in hereditary information. Otherwise, the molecule has, if it didn't have those variations, it wouldn't have any mutagenic properties. So by having variation, in other words, the molecule does have mutagenic properties. And the reason why that is important is because it creates genetic diversity within a species. And it also allows for the creation of different species, for example, speciation. And we know that genetic diversity is important because that allows for adaptation during stochastic events or stochastic change. So both of those characteristics are, again, necessary for heredity and necessary for DNA to be considered a hereditary molecule. Now, the set of genes in our DNA, or the inheritable information that's carried by all living things, that's known as an organism's genotype. Genotype holds the critical instructions that are used and interpreted by a cell's machinery to produce a trait. The physical expression or characteristics of said trait is the phenotype. So an organism's phenotype, or its traits and characteristics, are determined by its genotype, its genetic makeup, or the set of genes it contains. And as we talked about in some of the earlier lectures with evolution, there's also an environmental component to this, where the actual expression of phenotype is determined by this interactions between genotype and environmental conditions. And that based on certain environmental conditions, you could have kind of a different form of how that genotype is expressed to create this norm of reaction. Now, the building blocks of DNA are called nucleotides. You should recall this kind of from basic bio, kind of Bio 101, right at the starting line. DNA is known as a nucleic acid, right? That's what it's referred to as, and it's a macromolecule in that form. And in that form, it's also a polymer. Now, again, polymers are created by many monomers, so these nucleotides are the monomers of the polymer DNA. Now, the nucleotides themselves are composed of three parts. You have a nitrogenous base that's bonded to a pentose sugar. Pentose means it has 5 carbons, and that's pentose sugars bonded to the nitrogenous base by a beta N glycosidic linkage. And then you also have a phosphate group. Now, if you just had the nitrogenous base and the sugar components alone, they would form a nucleoside. The addition of that phosphate group gives you a true nucleotide. Now, there are 5 types of nucleotides. Identification as such is determined by the actual nitrogenous base. So there's 2 families of nitrogenous bases, pyrimidines and purines. Pyrimidines have a 6-membered ring of carbon and nitrogen atoms. Nitrogen tends to take up hydrogen ions readily from solution, hence the name nitrogenous base. As for your pyrimidines, you have cytosine, thymine, and uracil. Now, thymine is specific to DNA, uracil is specific to RNA. These are the guys we're talking about up here. So thymine's only in DNA, uracil is only in RNA. Now, the other group, your purines, they, on the other hand, are much larger. They're comprised of a 6-membered ring, but it's also fused to a 5-membered ring. Now, there's 2 types. You have adenine and guanine. Both of these are found in DNA. Now, how can you determine if a nucleotide is specific to RNA or DNA? And that's a very good question. The answer to that, as to whether the nucleotide is specific to RNA or DNA, really depends on the type of pentose sugar that's involved. That's this one here. The sugars differ from one another between the two. DNA has the sugar called deoxyribose. That's the sugar here, while RNA has ribose. Now, the 2 sugars differ from one another in regards to number and placement of the hydroxyl groups, particularly on the 1, 2, and 3' carbons. When we talk about the kind of the number prime carbon, we locate or designate those carbons moving in this clockwise movement. So this is carbon 1, 2, 3, 4, and then 5 is up here. So on the first, second, and third carbons, you have differences, again, in the number and the placement of the hydroxyl groups. In deoxyribose, in ribose sugars, you have 3 hydroxyl groups, one on the first, one on the second, one on the third. In deoxyribose, you only have 2. The second prime carbon lacks that hydroxyl group, or it lacks an oxygen atom. And that's what gives it its name in the first place. The name literally means 1 oxygen removed. So that's how you differentiate whether this nucleotide that's in question is DNA-based or RNA-based. Now, when we're looking at the actual structure of a DNA double helix, we can say that cellular DNA molecules have 2 polynucleotides, or strands of many nucleotides, that spiral around an imaginary axis. And that's what produces that familiar double helix model. In this model, adjacent nucleotides are joined together by phosphodiester linkages. Right, so these are the linkages here in between your adjacent nucleotides. And that is a type of covalent bond. That bond is more specifically formed by dehydration synthesis. And we talk about that in macromolecules. So if you need a kind of refresher on that, go back to the second lecture in the macromolecule series. But in particular, what's happening is the hydroxyl group on the third carbon of one nucleotide is joining with the fifth carbon of the other, and that creates this strand of repeated sugar and phosphate molecules, more specifically known as the sugar-phosphate backbone. Each backbone has two different ends. One end has the hydroxyl group on the 3' or the third carbon. This is known as the 3' end. The other end contains a phosphate group that's attached to the fifth carbon. Right, this is the 5' end. Now, when we talk about the two strands of the double helix, right? So here's the sugar phosphate backbone of one strand. Here's the sugar phosphate backbone of the second strand. These strands of the helix run in opposite 5' to 3' directions, which is an arrangement that's called antiparallel. So what that means is one strand of the helix runs 5' to 3', and the other is antiparallel to it, running 3' to 5'. Now, the structure itself of the entire helix is incredibly stable. And why do you think that is? Well, the reason for such is because of hydrogen bonding between the bases. This is what holds the two sugar phosphate backbones and strands of the helix together to maintain its shape. So to the outside of the helix, you have your sugar and phosphate groups. And then the nitrogenous bases of the nucleotides are positioned inside the helix, almost like rungs on a ladder. And they're done that, or they're positioned that way because of their hydrophobic behavior, right? Meaning they act to avoid extracellular fluid. So these bases are joined to the corresponding nitrogenous base by hydrogen bonds, and that forms a base pair. And the joining of these bases are dictated by base pairing rules. Now, with DNA, base pairing always occurs between a purine and a pyrimidine. So adenine always base pairs with thymine, and cytosine always base pairs with guanine, and vice versa. So the hydrogen bonds provide stability, right? So they stabilize the bases. With your adenine and thymine, there's always two hydrogen bonds that form between those bases. 3 will form between cytosine and guanine. That additional hydrogen bonding in your CG base pairs is what causes the DNA segments that have a really high GC content to be very difficult to denature. So you'll usually see the DNA of different organisms or different cell types that go through higher rates of DNA replication. They'll have more AT or TA base pairs because they need to readily go through replication, and therefore you need to have the separation or have these two strands readily separate in DNA to allow that. Cells that go through less replication and again need to be at a very high state of stabilization to kind of endure very extreme habitats or to avoid denaturing, they will have more CG base pair content. Now there's also vertical forces that are acting on the bases. So your hydrogen bonds are the horizontal. The vertical forces that are acting are your van der Waals interactions. They'll occur between the stacked bases. Now if we were to take one strand, right? So if we were to separate this and say we wanted to look at just this one strand and kind of stretch it out, we would be able to read the sequence of bases along that strand, and we would know the base sequence of the other strand as well, because the strands are complementary to one another. So they're anti-parallel and they're complementary. Again, that complementary behavior is due to these base pairing rules. Right? Thymine base pairs with adenine, or adenine base pairs with thymine. Guanine base pairs with cytosine, or cytosine base pairs with guanine. Right? So if you have thymine on your one strand, you know that the complementary strand has to have adenine at that same position. So if you were to essentially stretch out a strand of DNA that had a base sequence of A, G, G, T, C, C, G that ran from 5' to 3', then the base pairing rules would tell us that the other strand would have the sequence T, C, C, A, G, G, C running in the opposite way, 3' to 5'. So each strand becomes a predictable counterpart of the other. And this is the feature of DNA that makes it possible for precise copying of genes. Now, when we talk about the Watson and Crick DNA model, kind of the model or the go-to classic model of DNA, the two strands of DNA are twisted around each other to form a right-handed helix. Your helices do have a handedness to them, kind of like your enantiomers. You can imagine wrapping your right hand around the DNA molecule with your thumb pointing upwards. Now, if you rotated your hand clockwise to follow the turn in the backbone of this model, your hand should be moving upwards with the spiral in the direction your thumb is pointing. So because your right hand moves in the same direction as the thumb is pointing, the DNA double helix is right-handed. Right? So you're able to follow it with your right hand. That's a right-handed helix. If you were trying to do the same thing with your left hand, with your thumb pointing up, your hand would instead move downward, which is the opposite direction that the thumb is pointing. So again, all helices have this handedness to them, and this is important because it describes how their grooves are oriented in space. Your helices have minor and major grooves, right? The twisting of the DNA double helix and the unequal spacing of the sugar-phosphate backbone create these grooves, and they will run the length of the DNA molecule. Now the strand backbones, kind of what I'm referring to by this unequal spacing of the sugar-phosphate backbone, the backbones are closer together on one side of the helix than on the other. Again, that's because of this unequal spacing. And that results from the kind of geometrical orientation of the base pairs across the helix. So the kind of the takeaway, without going again too in depth with kind of the genetics of it, the grooves are important because they make the DNA molecule more flexible and accessible for interactions with proteins and other complex molecules. The major grooves in DNA can bind proteins. So the major grooves provide rich genetic information because they are, or because the bases there are more accessible. So again, this is an important property of DNA, an important property of its structure, because those major grooves, kind of in the upstream regulatory regions of a gene, will bind your transcription regulatory proteins. And just kind of a fun fact, some of your principal decoding mechanisms will rely on the ability of amino acid side chains to kind of protrude or move into those major group areas and recognize and bind to specific DNA sequences there. Now, again, all helices are not right-handed. Double-stranded DNA actually comes in 3 different forms, known as aDNA, bDNA, and zDNA. So although A and B DNA are both right-handed helices, zDNA is left-handed. So kind of with the putting your line of sight along the helices axis, if you have a clockwise screwing motion that moves the helix away from the observer, it's this right-handed helix. If it moves towards the observer, you have a left-handed helix. All right, so that's kind of the icing on the cake material, the basics for DNA. Let's move into its close friend, its little neighbor, RNA, right? Because we have 2 types of nucleic acids, DNA and RNA. RNA is the other type. RNA stands for ribonucleic acid. This is a molecule that plays many roles as far as the storage and transmission of genetic information. And these molecules are mainly involved in the process of protein synthesis, known as translation, and its regulation. Now, chemically, RNA is a polymer, or a long chain of nucleotides that are linked together, also by phosphodiester linkages, just like DNA. So that those bonds occur between the 3' hydroxyl group of 1 nucleotide or nitrogenous base and the 5' or the phosphate group on the 5' carbon of the other. Now, unlike DNA, your RNA is primarily single-stranded. There's some double-stranded RNA, but most of it is single-stranded. And the nucleotides that are found in RNA are specific to RNA. We call them ribonucleotides. Again, very much like your nucleotides, they consist of 3 parts. They have a sugar, the pentose sugar that's specific to RNA is ribose. They have a negatively charged phosphate group, and then one of 4 nitrogenous bases, kind of attached at the 1' position. So these nitrogenous bases, they are all the same as DNA, except you do not use thymine. Instead of thymine, you use uracil. So again, thymine is specific to DNA, uracil is specific to RNA. Otherwise, all of them are the same. So for RNA, you have adenine, guanine, cytosine, and uracil. And the base pairing rules are the same as DNA. So you always have a purine base pairing with a pyrimidine. So A base pairs with U, C base pairs with G, and vice versa. Again, your RNA-specific pyrimidine uracil will form that complementary base pair with adenine and is used instead of thymine, which is specific to DNA. Now the sugar that's used in RNA, again, is your ribose sugar. The sugar ribose has a hydroxyl group that's attached to the pentose ring at the 2' position. So it's attached to the 2' or the 2nd carbon. This hydroxyl group placed here it has a total of 3 hydroxyl groups. But more specifically, this hydroxyl group is what makes RNA less stable than DNA because it's more prone to hydrolysis, which is the cleavage of chemical bonds by the addition of a water molecule. So this makes RNA more suitable for its short-term functions, right, as opposed to DNA, which is better for long-term storage of genetic information. It also causes RNA to adopt a different type of geometry. That's called an A-form geometry, which influences those major and minor grooves. And again, the base pairing rules will be the same as you see in DNA, except that instead of using thymine, you have uracil. Right? Uracil is essentially this unmethylated form of thymine, but that's the kind of the complement for adenine that's specific to RNA. Now there's several different forms of RNA, each with its kind of own unique function. mRNA, that's kind of serves in creating the amino acid of a polypeptide that's made from protein coding genes. There's RNA, tRNA, you have snRNA. These occur only in your eukaryotic genes, and they participate in mRNA processing, more specifically in the removal of introns. There's miRNA, which is microRNA, which are only found in plants and animals. They're involved in post-transcriptional regulation of mRNA through what are called RNA interference mechanisms. And then there's siRNA, which are your small interfering RNAs. Again, they're also only found in plants and animals. They provide protection against viral attacks and kind of minimize the spread of transposable elements. The major ones that I want you to focus on are mRNA, tRNA, and rRNA. So let's go a little in depth with those three forms. Kind of starting from the very beginning, cells access the information that's stored in DNA by creating RNA to direct the synthesis of proteins through the process of translation. Translation, again, is protein synthesis, or the creation of proteins. Now, the three main types of RNA that are directly involved in protein synthesis are your messenger RNA, which is mRNA, ribosomal RNA, which is rRNA, and transfer RNA, which is tRNA. So that's why we're focusing on these three types. Now, mRNA serves as an intermediary, per se, between DNA and its protein products. What happens is information from DNA is transcribed into a strand of mRNA through the process of transcription. And that mRNA then carries that genetic information or genetic message out of the nucleus and into the cytoplasm for synthesis. So if DNA serves as kind of the complete library of cellular information, you can think of mRNA as a photocopy of specific information, again, serving kind of as the blueprint instructions for making a protein. You can also think of it as making a photocopy of a recipe that you have to bake a cake, and your friend wants that recipe. So you don't get for the original recipe, because she's notorious for losing things, and then you'd never have that recipe again, so you wouldn't be able to make that fantastic Victorian sponge cake. So you make a photocopy of it, right? You give your friend the photocopy, because if she loses it, you still have the original. Right? You're not kind of the sad man out in that case. So that photocopy, think of as mRNA. The original template is your DNA. Now, if a cell requires a certain protein to be synthesized, the gene for that product is turned on, and then your mRNA is synthesized. So that's mRNA. Ribosomal RNA, your rRNA, is a relatively stable type of RNA that is the major constituent of ribosomes. They actually compose roughly 6% of its mass. The rRNA in the ribosome is the catalytic enzyme that will read the strand of mRNA and synthesize the polypeptide chain from it. But the RNA in that ribozyme, or your actual ribosome, ensures the proper alignment of the mRNA to the ribosome during protein synthesis. And again, it also catalyzes the formation of the peptide bonds between amino acids during protein synthesis. And that's what allows you to create the polypeptide unit, a chain of many amino acids. And then there's transfer RNA, or tRNA. This is kind of the third main type of RNA that we're talking about. It's one of the smallest, usually only 70 to 90 nucleotides long. And like RNA, it's one of the stable types of RNA. In both prokaryotes and eukaryotes, your tRNA is encoded kind of in the DNA and then copied into long RNA molecules that are then cut to release smaller fragments containing the individual mature RNA species. Now, your tRNA will come into play during protein synthesis in that it contains what are called anticodons. So these are the complement codon sequence of the codons in mRNA. So if you had a codon in mRNA of, we'll say, UUC, then the anticodon of the complement tRNA would be AAG. But when the correct tRNA for that codon base pairs to the mRNA strand, it brings in an amino acid. So right at the top of the kind of 3D structure of the tRNA molecule is there's an amino acid that's linked to it. The amino acid corresponds to the mRNA codon. So that's how these amino acids are kind of shuttled into the ribosome. The ribosome, using its rRNA, will link the amino acids together to form the polypeptide chain. And again, that sequence of amino acids is determined by the codon sequence of your mRNA. So those are the kind of, again, three major types of RNA. And kind of as for the overall basics on DNA and RNA, that's kind of a good starting point as far as a foundation for you, without going too far in the nitty-gritties. So if you have any questions, feel free to leave them in the comments section, or you can get a hold of my email and email me directly. But I hope that this helped you understand kind of the basic information as far as the structure and function of DNA and RNA. And as always, I hope that you learned something that you didn't know before, and you're walking away with a clearer understanding of the material.